Hello everyone and welcome to my channel Being Biotechnical. So, in this series of videos, we are discussing about structural basics of DNA and RNA. And this is the part 3 of this series of video lectures. In the previous lectures, we already have discussed about the components of nucleotides, and we also learned how to draw their structures in simplified way. If you have yet not seen the previous part of this topic, I would recommend you all to please do go through them once for understanding the whole concept clearly. So, without further ado, let us jump into the topic. So today, we would see how the nucleotides are joined together and what is a polynucleotide. So, nucleotides are composed of three components. First is nitrogenous base, second is pentose sugar, and third is a phosphate group. In the previous videos, I have already discussed their structures with you. Now, let us see how they are joined to one another to form a nucleotide. So, let us first draw a pentose sugar. This is the structure of a pentose sugar. We have to place this pentose sugar by facing the oxygen towards the left side, which is like this. Okay? So, this pentose sugar is attached to the base. This pentose sugar is attached to the nitrogenous base. Let us consider this is a nitrogenous base. We can also write it as simply as base. So, this base is attached to the first carbon of the structure. Now, which is the first carbon of the structure? This is the first carbon of the structure, because we have faced the structure of the pentose by facing the oxygen towards the left. So now this becomes the position of the first carbon. This nitrogenous base would be attached to the first carbon of the pentose sugar. So let us join them. So I have highlighted the bond with red color. As you can see, I have highlighted this bond by which the nitrogenous base is attached to the pentose sugar with red color. This is because this bond is an N-glycosidic bond. So in order to draw your attention towards this, I have highlighted this with red color. So always remember that this nitrogenous base is attached to the pentose sugar with the help of an N-glycosidic bond, which I have marked in red color. Okay? So, remember, this is a nitrogenous base, this is a pentose sugar, and the bond by which they are connected is an N-glycosidic bond. One more thing you need to remember is that pentose sugar, when attached to the nitrogenous base, will together be known as nucleoside. So, remember, pentose sugar plus nitrogenous base will together be known as nucleoside, or pentose sugar plus nitrogenous base will give you nucleoside. They both, when joined to one another, is known as nucleoside, which means this is now the structure of a nucleoside. Clear? This is now a nucleoside. But when this nucleoside will be attached to the phosphate group, then the whole structure would be known as nucleotide. So remember, nitrogenous base plus pentose sugar is equal to nucleoside, and nitrogenous base plus pentose sugar plus phosphate group is equal to nucleotide. Okay? So for your better understanding, just look at this image. Look, this is pentose sugar. This pentose sugar plus nitrogenous base is equal to nucleoside. This nucleoside plus phosphate group is equal to nucleotide. Okay? let us come back to our structure. So, see, this is the structure of a nucleoside, which is pentose sugar and nitrogenous base are now joined by this N-glycosidic bond. They are both now joined by this N-glycosidic bond. Now we have to add phosphate group to this structure in order to make this the structure of a nucleotide. So this is a phosphate group. We may denote it by P. Okay. Now this phosphate group would be attached to the 5' carbon of this pentose sugar, which is CH2OH. 5' carbon is this CH2OH. Okay. So now let us join this phosphate group to the pentose sugar. Done. The reason why it binds to the 5' carbon of the pentose sugar is that the valency of carbon is 4. And look at this carbon. It has 2H and 1 OH attached to its structure. So this carbon is not stable because 2 hydrogen and 1 OH group is attached to it and the valency of carbon is 4. So this carbon was not stable. It still needs to get attached to one more compound in order to be stabilized. So that's why this phosphate attached to this carbon of the pentose sugar. During this process, what happens is that when the phosphate comes in close proximity to the 5' carbon of the pentose sugar, then these 3 OH groups react and one water molecule is released. So now look at this structure. This phosphate, when comes in close proximity to the pentose sugar, then this OH and this OH, which is 2 OH of this phosphate group and 1 OH of this pentose sugar, they will react and one water molecule will be released. Okay. So after the releasing of this one water molecule, what will happen is this phosphate group even becomes negatively charged. So as the water molecule is now released, so this phosphate group is now negatively charged, which is why this phosphate group shares this oxygen with this carbon, with the 5' carbon of the pentose sugar. So always remember then that whenever a phosphate is attached to a carbon, then they are both attached by phosphodiester bond. I have already marked this bond by green color. So this green color bond is a phosphodiester bond. Whenever a phosphate group is bound to a carbon, then they are both bound to one another by a phosphodiester bond. Okay. Now this whole structure is a nucleotide. Now this whole structure is a nucleotide. Similarly, when many nucleotides are attached to one another, as we saw that this is the structure of one nucleotide, so similarly when many nucleotides will attach to one another, then that will be known as polynucleotide. That will be known as polynucleotide. Poly means many. Now, how are many nucleotides joined to one another? So many nucleotides are joined to one another by phosphodiester bond. Now the question is how? So look at this carefully. This is the structure of a nucleotide and this is the structure of another nucleotide and so on. This is one nucleotide, this is two nucleotide and so on. These nucleotides will be joined to one another by phosphodiester bond. Look at this carefully. Join this phosphate group with the 3' carbon of the successive group. Okay? Do this to all the nucleotides. That is, join this phosphate to the 3' carbon of the successive nucleotide. You will now see that phosphate is linked to all the nucleotides at 2 positions. You will now see that phosphate is linked to all the nucleotides at 2 positions. One is 5' and another is 3'. And in both the sides, the phosphate is linked with the help of phosphodiester bond. Okay? In both the sides, the phosphate is linked to the structure by the help of the phosphodiester bond, which is this is also a phosphodiester bond and this is also a phosphodiester bond. Carbon and phosphate are always linked to one another by phosphodiester bond. I want you all to focus on the structure once again. See, these are single nucleotides. Okay? These are single nucleotides. These single nucleotides are joined to one another by phosphodiester bond at 5' and 3' ends. Okay? Done? Now you will find that at the left side, there is a phosphate group which is not attached to any nucleotide. Right? This is the phosphate group which is not attached to any nucleotide. On the right side, there is no phosphate groups attached to the 3' carbon. So this is the right side, this is the 3' carbon, and see here no phosphate group is attached. Okay? So we may also say that this phosphate is free and this 3' carbon is also free. This phosphate group is free and this 3' carbon is also free. As this phosphate is attached to the 5' end and is free, and the last nucleotide is also free, then they both will be known as the 5' end of the polynucleotide chain and the 3' end of the polynucleotide chain. So this is how we name the ends of the polynucleotide chains as 5' end and 3' ends. I hope you are all clear about the structure of polynucleotide chain. In case of DNA has two nucleotide chains. DNA has two nucleotide chains arranged in this form. One is present from 5' to 3' and another is present from 3' to 5'. We may also say that both the chains are arranged anti-parallel to each other. And these two polynucleotide chains are arranged like this. Now, look at them carefully. This is one nucleotide chain arranged from 5' end to 3' end, and this is another polynucleotide chain arranged from 3' end to 5' end. We may also say that these two chains are arranged in anti-parallel to one another. These two chains are arranged anti-parallel to one another. So, the one in green color are the phosphate groups and those in red color are the nitrogenous bases. We have already discussed that nitrogenous bases are of two types, purine and pyrimidine. The purine will always be attached to the pyrimidine in a polynucleotide chain. The purine will always be attached to the pyrimidine in a polynucleotide chain. So for your better understanding, look at this figure. Purine and pyrimidine, which are both the components of nitrogenous bases. So adenine and guanine are both purines. Cytosine, thiamine and uracil are the three components of pyrimidine. You may also recall it as pure and good. Pure means purine, take A from this end and A for adenine, then take G from this good and G for guanine. So clear? Pure and good means purine have two things, purine have adenine and guanine. Okay, purine have two components, adenine and guanine. Similarly, remember for pyrimidine that Python can't trouble you. Python can't trouble you. P means pyrimidine, C from can't, C means cytosine, T from trouble and T for thymine, and take U from U. U for uracil. Okay? So, cytosine, thymine and uracil are pyrimidine. Okay? By this way you may recall it very easily. One more thing you need to recall is that always remember that thymine is only present in case of DNA and uracil is only present in case of RNA, which is when it comes to DNA you will find adenine, guanine, cytosine and thymine. But when it comes to RNA you will find adenine, guanine, cytosine and uracil. So uracil is not present in DNA and thymine is not present in case of RNA. Clear? That is why I have marked this DNA and RNA against their names. Okay? One thing you also need to remember that adenine will always bind to thymine and guanine will always bind to cytosine. So A will always bind to T and G will always bind to C. ATGC. Okay? Now let us return back to our structure. So these are the places where we have to fill the nucleotide bases in order to make this the structure of a double helical DNA. So look carefully. C, G, A and T. Again T, A, C and G. Double bond, triple bond, triple bond, double bond. This is how DNA is arranged in a double helical structure. And in the similar way, when uracil is placed in place of thymine, then that is the structure of an RNA. Look at this. C, G, A and U. Okay, this is the structure of a RNA. RNA usually are single-stranded in structure. So with this, the structural basics of DNA and RNA is done. If the video was useful to you, then please do hit the like button, share this video with your friends, don't forget to subscribe to my channel and do hit the bell icon for further updates on my channel.